Essentially, there are two types of cells that exist in nature, prokaryotic and eukaryotic cells. The first that we're going to discuss in this class is going to be the prokaryotic cell. Prokaryotic cells are the most abundant and diverse life forms found on Earth, and they're found virtually everywhere, in the air, in the soil, in the water. They're also found on or in living organisms. Some of the key characteristics of prokaryotic cells is that they lack a membrane-bound nucleus or organelles. They tend to be structurally smaller and simpler than eukaryotic cells. And the prokaryotic cells are going to be placed within two taxonomic domains. Bacteria can cause diseases, but they are also environmentally extremely important as decomposers. They also play a very important role in our own guts, producing a lot of the vitamins and nutrients that we can't produce on our own or obtain from our surroundings. They can also be very useful in manufacturing products and drugs, as we will discuss in a later chapter. Prokaryotic cells are also found in the domain Archaea. These are going to be our extremophiles. They're going to be found living in the most extreme habitats. These two prokaryotic domains are structurally very similar, but biochemically they are extremely different. So we're going to start off by talking about the structure of prokaryotic cells. Like I said, they're pretty small. These are going to be extremely small, at least compared to the eukaryotic cells. They're going to be between 1 and 2 micrometers wide and maybe 2 to 6 micrometers long. They're going to occur in 3 basic shapes. The spherical, also known as coccus, the rod shape, known as bacillus over here, and then the spiral or the spirillum if they are going to be rigid, or spirochetes if they're sort of a little bit more flexible, like a spaghetti noodle. Now the structure of the prokaryotic cell, like I said, it is relatively simple, but it does contain a few key characteristics that we are going to go over. The very first feature of the prokaryotic cell that we're going to talk about is the cell envelope. The cell envelope is going to include all of the structures that you see here: the plasma membrane, the cell wall, and then the glycocalyx. The first step being the plasma membrane. Sorry about that, it should say plasma membrane. Next, the cell wall, and then finally the glycocalyx. We are going to go over each one of these. So starting off with the plasma membrane. The plasma membrane is going to be a lipid bilayer that has both embedded and peripheral proteins. As we previously mentioned, this lipid bilayer is going to be composed primarily of phospholipids. And if you remember from our talk, we said that one region of the phospholipid is going to be polar and the other region is going to be nonpolar. Now the polar regions are going to have an attraction for water. They're going to be hydrophilic. The nonpolar regions will be hydrophobic. Because of this, the polar regions are going to orient themselves to the outside of the cell and the inside of the cell, where water is going to be abundant. The nonpolar The nonpolar tails are going to be found on the in between those two polar regions. So the plasma membrane is going to be a lipid bilayer with embedded and peripheral proteins. The next layer we're going to come across is going to be the cell wall. The cell wall is going to help maintain the shape of the cell, and it's going to be strengthened by peptidoglycan. If you remember, peptidoglycan is going to be the structural carbohydrate found in bacteria. The outermost layer of the prokaryote is going to be the glycocalyx. That's going to be this thick layer that you see out here. Now this is going to be a layer of polysaccharides. So again, these are going to be carbohydrates that are found on the outside of the cell wall. In the event that the glycocalyx is a well-organized and resistant to removal, it's known as a capsule. If you can wash it off easily, it's known as a slime layer. Its primary feature, or its primary function, is going to be to prevent the bacteria from drying out in drought-like conditions. It also helps bacteria to resist host's immune system. It helps with adhesion to surfaces, and it helps with adhesion to surfaces. So it does serve several purposes. That's going to be the outermost layer, or the cell envelope found on the outside of the prokaryotic cell. Moving inward, we're going to come across the cytoplasm. The cytoplasm is going to be a semi-fluid solution that's ultimately encased by the plasma membrane. It's going to contain water, inorganic and organic molecules, and enzymes. Inside of that cytoplasm, we're going to find what is known as the nucleoid region. The nucleoid is a region that contains the single circular DNA molecule. Bacteria, and archaea, so prokaryotes, only have one chromosome. They have one great big long circular DNA molecule that's going to contain all of their genetic information. Now, they don't have a membrane-bound nucleus where they can store and protect their genetic information, but they do have a region where they generally keep their single circular DNA molecule. Eukaryotes, on the other hand, can have multiple chromosomes, but prokaryotes will only have that single coiled chromosome. Now they only have that single coiled chromosome, so that chromosome is going to encode for everything that bacteria needs in order to be a bacteria. However, bacteria do have this very unique feature where they can actually have what are known as plasmids. Plasmids are going to be small accessory rings of DNA, meaning they're extra chromosomal. They're not part of the main genetic code. They're not part of the chromosome, but it's extra chromosomal outside of that chromosomal DNA that the bacteria might find useful. These plasmids are often going to be used in biotechnology laboratories as a vector to transport DNA into bacterium. And we're going to have a whole section on this in chapter 14. It's a very exciting branch of science. Next up are going to be the ribosomes. Ribosomes are going to be found studded all throughout the cytoplasm. These are tiny structures in the cytoplasm. Their ultimate goal is going to be to synthesize proteins. So, like we mentioned earlier, nucleic acids such as DNA and RNA are excellent molecules for genetic storage, but storing DNA does very little good if that information can't be turned into something useful. It's kind of like your textbook. There's a lot of great information in there, but unless you open it up, read it, and apply that information, it does you no good. And so that's where the ribosomes are going to come in. The ribosomes are going to be responsible for reading the genetic information stored inside the DNA and turning that genetic information into proteins. So ribosomes, you'll also see them referred to as protein factories because of their role in producing proteins. Finally, we're going to talk about the external structures found on the outside of the prokaryotic cell. So for example, there is going to be the flagella. Not all prokaryotic cells will have this, but some will. The flagella is going to be there to provide motility. The external structures of the prokaryotic cells are going to be typically made of proteins. Motile prokaryotes can propel themselves through water with the help of a flagella, and that flagella is going to actually be able to rotate a full 360 degrees, kind of like a boat motor. So it actually will rotate in a full circle. There may also be structures known as fimbriae. Fimbriae are going to be these small bristle-like structures that you see right here. They're going to sprout from the cell surface. Some prokaryotes have fimbriae that are going to sprout from that surface. They do not play a role in locomotion. They're not cilia. They're not going to beat back and forth. They are not responsible for moving the cell back and forth. They are used to help attach to surfaces. You can kind of think of this as sort of a Velcro outside layer. It's not going to be useful for movement, but it will help it adhere to surfaces. And then finally, there's going to be the conjugation pili. The conjugation pili is going to be a rigid tubular structure that is used to pass DNA from cell to cell. A singular form of that would be called a pilus. Now, as we have mentioned sort of very briefly in chapter 1, there are some organisms that reproduce asexually. They don't actually share genetic or swap genetic information in order to reproduce. And a great example of that would be prokaryotes. Prokaryotes do reproduce asexually, but even though they reproduce asexually, they do have the ability to actually share DNA. Specifically, they're going to share not their entire chromosome, but they're going to share those tiny little extra chromosomal plasmids that we mentioned earlier. So, for example, if one bacteria picks up a bit of extra chromosomal DNA, a plasmid that has the genetic information to be resistant to an antibiotic, it can make that antibiotic resistance. It can then then actually pass it on and share that information with all of its neighbors. So not only will it pass it on to its offspring, but it can share that extra chromosomal DNA with all of its neighbors, and suddenly we have an entire colony that's going to be resistant to antibiotics. So it's a very interesting process. Now we can use this to our advantage, and we'll talk about that in our biotechnology section of this chapter. All right, that's it for our discussion on prokaryotes. Please let me know if you have any questions, comments, or concerns.